Hoje temos a presença de uma convidada internacional para esse webinário. Tá? Eu vou, então, apresentá-la em inglês, bem como o webinário será em inglês dessa vez. Qualquer dificuldade ou dúvida, por gentileza, não hesitem em usar o chat, ou mesmo ao final da apresentação, perguntas podem ser feitas em português, que nós traduzimos, né? Bem como as perguntas do chat também. Qualquer dificuldade no entendimento, nós estamos à disposição para ajudar nesse entendimento, para fazer a tradução se necessário, ok? Bom, com isso dito, eu vou então apresentar a nossa palestrante de hoje. E a partir de agora, eu vou falar só em inglês, ok, pessoal? Então, vamos lá. Well, hi everybody. Welcome again to our monthly webinar. Today we have a very special guest, an international guest, Dr. Bajia Burla. Dr. Burla has a PhD from the Iowa State University from 2017, where she developed a work focused on next generation sequencing technologies. Since then, she established a career in genomic products and services and landed a job at GENEWIZ, where she provides technical consultations for you guys, for our projects, for customers that desire to design their NGS projects and make scientific recommendations as well. In other words, she will support you to design, establish, understand your NGS project, right? So she definitely has a lot of experience to share with us today from her expertise in the field, and well, definitely the right person to talk about your DNA RNA sequencing project, right? And that's the kind of support and deep knowledge that we will find at GENEWIZ. That's why GENEWIZ is now our partner and offers these services, among others, that she will talk about briefly. And well, without any further ado, I'd like to welcome Dr. Burla. It's a real honor, and please, you have the word. The stage is yours now. Feel free to share your screen, and thank you very much for your time.
Thank you, Douglas, for the kind introduction. Hello, everyone. I'm Bhagya Birla, and I'm happy to be here with you today and talk about our next generation sequencing services. So I'm sharing my screen now. Can you see my screen?
Yes, we can.
Okay, perfect. All right. So as Douglas mentioned, the topic of my talk today is the next generation sequencing solutions that we offer here at GENEWIZ. So in general, the outline for my presentation today is to give you an overview of some of the different services that we have at GENEWIZ, along with a deeper dive into some specific next generation sequencing services, including RNA-seq, whole genome sequencing, and microbiome sequencing. So as GENEWIZ, I'd like to give you an overview of who we are and what we do. So GENEWIZ is a global services company. We provide genomic services in many different categories, including Sanger sequencing, gene synthesis, next generation sequencing, as well as preclinical and clinical services. So together with all of these technologies, we like to say that we can do both reading genes using sequencing technologies such as Sanger or NGS, as well as writing genes using synthetic DNA technologies. All of these encompassing together have a wide variety of applications in many different areas, such as drug discovery and development, infectious disease, single cell research, biomarker discovery for diseases, synthetic biology, and many, many more. We are a global company with our headquarters based in New Jersey, and all of our customers based in North America and South America send samples to our lab in New Jersey for processing. We also have NGS labs based in other parts of the world, including a lab in Germany for our European customers. And if you have any collaborators who are based in Asia, we have 2 NGS labs in Asia, in China and Japan. So I'd like to talk about what exactly the workflow looks like for our next generation sequencing customers. We have a complete array of services, right from project consultation up to data analysis and delivery support. Initially with our PhD level scientist team, we would consult for your particular project and be able to guide you for your project design so that you're able to make the best possible project goals and project design to be successful with your experiment. We can do the nucleic acid extraction for you, or you can do this yourself and ship the extracted DNA or RNA to us. And GeneViz will provide the next generation library preparation and sequencing services, as well as bioinformatics if you need it, or we can just deliver the raw FASTQ data with you and to you and guide you in terms of what kind of analysis may be needed. In case you decide to proceed with bioinformatic analysis at our end, we also offer post-delivery support, which includes scheduling another consultation to go over any of the results that have been generated for your particular project and data. All of our workflow is managed through an integrated customer laboratory management software system, and this is available online. You can open or register for an account freely, and this account can be used for any of our downstream workflows for NGS. This system can be used for requesting a quote, confirming an order, and as well as tracking the project status. So once you do register an account with us, you can do all of these different things using your online account. So we do know that you may have a lot of other options out there for next generation sequencing, and there may be other providers out there, but I'd just like to talk about why you should choose GeneViz as your NGS provider, as we provide the best overall value for your research needs. With GeneViz, you have many different value services that are included along with your next generation sequencing workflow. We help you order and track your project completely online. We offer guaranteed turnaround time for some of our sequencing packages, as well as dedicated project managers who will communicate with you at various stages of the workflow. We offer guaranteed data quality for the results for the sequencing data, and also include sample QC in our turnaround times. So we know that a lot of other providers out there may not include sample QC in their particular turnaround times that they provide you along with the quote, but we like to be as transparent as possible, and we will give you the exact turnaround time that would be needed to complete your project without without hiding the sample QC times or anything like that. We also have many different packages depending on your particular project deadlines and budget requirements. Some of these packages include value, preferred, and express services. So depending on your particular budget, we can work with you to figure out which package service might be the best for you. The value package is going to be the most economical one, whereas the preferred and express options are offered in case you have a project deadline or a grant due and you want to get your data as soon as possible. So now I'd like to move on to talking about the various next generation sequencing services that we offer. You may have used other molecular biology techniques for your experiments before, such as qPCR or microarrays. And with NGS, you actually get a much higher resolution of detection of DNA or RNA in the samples. And we offer a wide variety of services depending on your particular research goals and applications. So we have services that include amplicon sequencing, whole genome sequencing, targeted sequencing, epigenomics, metagenomics, and so on. For today's talk, I'll focus on few services as that were requested for the group here. At GeneWiz, we have a couple of different platforms for sequencing. We have the Illumina, HiSeq, MySeq, and NovaSeq platforms for short-read sequencing. And we also have the PacBio Sequel 1 and Sequel 2 platforms in-house for if you require long-read sequencing technology for your projects. We also have the 10x Genomics Chromium platform if you are working with single cells and want to outsource any of your single cell RNA sequencing or ATAC sequencing projects. So now I'll move on to talking about specifically RNA sequencing service offerings at our end and some of the applications that you could use RNA sequencing for. So this type of NGS or RNA-seq can be highly useful in gene expression studies between treatment and control samples. So for example, if you have a particular, if you're studying particular cells and are interested in diagnosing the cell response studies to particular treatment, disease state, infection, or any other condition, we can generate a gene or transcriptomic profile of that particular disease state or treatment cell sample condition, along with the control sample to generate a comparative transcriptomics as seen on the right here, and give you a gene expression profile of the control versus treatment group samples. RNA-seq can also be applicable in biomarker discovery, single-cell transcriptomics, de novo transcriptome assembly, as well as small RNA and microRNA studies. We offer multiple workflows to fit your particular individual project goals. So we have say 3 different types of library prep methods for the type of experiment that you're designing. So for example, if we start with RNA extraction, we can do either poly-A enrichment if you only want to look at the messenger RNA that is present in the sample, that's mRNA, and we can do the mRNA library prep followed by sequencing on the Illumina platform, as well as provide the downstream bioinformatic analysis. And I'll talk a little bit more about the different types of bioinformatic analysis that we offer. The other workflow option would be ribosomal RNA depletion in case you want to analyze both the messenger RNA as well as long non-coding RNA that is present in your samples. That would be considered a whole transcriptome library prep, and this would again be followed by sequencing and analysis. Additionally, we also offer small RNA specific library prep. So in this case, if you're working with a specific microRNAs and you want to identify the microRNA profile in samples, this particular small RNA library prep option would be the most suited for you. Along with our standard RNA sequencing workflow, which requires a minimum RNA of 100 nanogram to start with, we also have the ultra-low input RNA-seq workflow, where we can work with as low as 10 nanogram of total RNA or as few as 1,000 cells to start with. All of these would be both ultra-low input as well as standard mRNA-seq would be a poly-A enrichment workflow. But if you are interested in studying specific bacterial samples, then ribosomal RNA depletion method would be more applicable. We also make certain specific read recommendations, that is the Illumina sequencing reads for particular organisms that you're working with. So if you're working with bacterial samples or small eukaryotic samples, we recommend 10 to 15 million head and reads per sample to get a good understanding of the gene expression profile of the samples. And if you're working with large mammalian genomes, then we recommend a higher number, that is 20 to 30 million head and reads per sample for a gene expression profile of those. Along with this, if you're working with any different kinds of species, we would be happy to schedule a technical consultation with you and provide guidance as to the best read recommendation for your particular sample type. Additionally, with the Illumina sequencing, we also offer RNA-seq on the PacBio platform, which is the long-read sequencing, and I'll talk a little bit more about that as well. As an added service to our RNA-seq, we also offer RNA stabilization tubes so that it is convenient for you to ship your RNA samples to our lab in New Jersey. What you need to do for this is just extract the RNA and add it to these stabilization tubes and follow a simple workflow to ship your RNA samples to us at room temperature without any additional cost of dry ice. We provide these RNA stabilization tubes as a complement, are complementary if you have a confirmed RNA sequencing project with us. Any RNA that is added to these tubes is stable at ambient temperatures for a long time. So as I mentioned, we have various different RNA analysis options with many customizable add-ons. We offer standard data delivery with QC report for read quality if you only need the FASTQ, raw FASTQ data to be delivered to you. We'll make sure that the QC report includes read quality metrics that meet our standard quality score guarantees. Additionally, with our standard RNA sequencing package, we would be able to provide gene expression analysis, splice variant analysis, and gene ontology analysis. If you need any additional data analysis more than that, we could do SNP indel detection, gene fusion detection, as well as de novo transcriptome assembly. And we have many different options for data delivery, including SFTP or hard drive delivery. But if you have a cloud server set up, we can also upload your data to that particular cloud server. So along with the Illumina short read technology, we also have the RNA sequencing available on the PacBio long read platform. One of the reasons that researchers decide to sequence RNA samples on the long read platform is because it overcomes the limitations of Illumina sequencing to identify alternatively spliced transcripts and novel isoforms. So with short read sequencing analysis, since the short reads do not cover the entire transcript, some of this analysis would be a predictive approach for splice variants. But with PacBio, since we are able to generate full-length cDNA sequence reads, we do not require any assembly or predictions, and we are able to identify the different isoforms that are present in the particular samples. I am now going to discuss our offerings for whole genome sequencing. As with RNA-seq, we provide both short read and long read technology for whole genome sequencing projects. And some of the applications for whole genome sequencing that may apply for your particular projects is if you're working to identify variants that cause a particular tumor or cancer, or genes that contribute to particular antimicrobial resistance in bacterial samples, or if you're performing certain de-extension or preservation studies. So with whole genome sequencing, with the Illumina platform, since it is a short read technology, we would need a reference genome to be available for the particular species that you're working with. to perform variant detection, that is identify single nucleotide variants, short insertion deletions, and germline or somatic mutations. We can also do de novo assembly of the particular genome, but we would require a reference genome for that. In case you do not have a reference genome available on NCBI because the species that you're working with is very rare, we can do the sequencing on the PacBio long read platform and help you with the de novo genome assembly of the particular sample. PacBio long read technology is also useful for discovery of large insertion deletions, translocations, inversions, and copy number variations, as well as haplotype phasing. We have certain standard requirements for genomic DNA to be submitted to us so that we can proceed with these workflows. The recommended amount would be at least 100 nanogram of starting DNA, but if we do have PCR-free library prep options available as well, for which we would require 1 to 2 microgram of DNA as a starting material. We also make some recommendations for sequencing of a particular species type on different platforms. So if you are working with very small genomes such as bacterial or smaller animals, we recommend sequencing on the MiSeq or the HiSeq platform. Whereas if you're working with larger genomes, we would recommend sequencing either on the HiSeq for fewer samples, or if you have a large number of samples, we would recommend sequencing those on the NovaSeq platform. Some of the advantages of long read technology as compared to Illumina short read is especially when you are trying to assemble a complete genome sequence. The Illumina short read, say you have a genome with a highly repetitive region or extreme GC content region. The Illumina short reads, when we look at them, they are not going to be spanning the entire repetitive region. They are only going to be spanning a particular short areas of that region, and hence we would not be able to get them to map back to the entire region, and this is going to result in large gaps in the assembled genome. With the long read technology, on the other hand, we would be able to generate reads that pretty much span the entire repetitive regions, and hence we would be able to provide coverage in this very difficult area of the genome. So this is a comparison between when we would recommend short read as compared to long read technology for whole genome sequencing. As I mentioned, short read technology would be recommended if you're looking at somatic or germline variants, copy number variants, or de novo assemblies of easier genomes that do not have as many repetitive regions. This is going to be more cost effective for you in terms of budget. If you do have a greater budget, or if you are looking for specific de novo assemblies of complex genomes or detecting large translocations or structural variants, then we would recommend going with the long read technology. So moving to microbiome diversity and some of the applications in this area. Microbiome sequencing has a wide variety of applications in many different fields like agriculture, industry, and it helps us to understand the world around us and are very good indicators of animal and human health. Lately, microbiome sequencing has been a main area of focus of many different labs, even if they have been working with other types of research like RNA or whole genome sequencing. Labs are also interested in assessing the microorganism diversity of the specific samples that they are working with to identify functional relationships. So for assessing microbiome diversity, we have two different solutions. One is a 16S Easy service, which is basically a genus level taxon identification and a targeted analysis. It is a fast and cost-effective service if you specifically just want to identify the different types of bacteria that are present in the samples. Along with bacterial identification, we also have a fungal assay that is developed to identify just the fungi that are present in the samples. As opposed to this, there's another solution, which is whole genome metagenomics, which can be used for subspecies level taxon identification and is ideal if the particular bacterial genomes have poorly annotated 16S regions and are helpful, and this technology is helpful to identify functional and genomic information. So diving a little bit deeper into 16S Easy service, we have developed a certain set of proprietary PCR assays for specific V3 and V4 regions of the bacterial genomes. These V3 and V4 regions are most variable regions between different bacteria, and hence once we are able to sequence these regions, we can identify the different types of bacteria that are present in the samples. With this service, we provide a complete KIME-based bioinformatic analysis report. And this chart here shows different samples and how the different bacterial, these different colors represent different bacterial populations that are present in the samples. And we see that there's a difference between the 4 samples that we are studying, and can then after this make educated interpretations of why there's a different bacterial populations being present in different samples can be it could be associated with specific diseases or particular environmental conditions, depending on the environmental or human or animal research that you're doing. With whole genome metagenomics, we are basically going to sequence the complete genome of the bacteria or any of the microbes that are present in the samples. We would, as opposed to 16S sequencing, which is a targeted approach, this is going to be sequencing the entire genomes. And thus, we would be able to identify not just bacteria, but also fungal, archaea, and viral species that are present in your samples. So if you're working with many different microbial species or are interested in identifying different microbes in the particular sample type, like environmental samples or gut microbiome, etc., then this would be the recommended approach for you. Whole genome metagenomics is also useful as it can help with taxonomic identification. So we can do a species level identification of the bacteria and also able to determine the functional relationships between microbe and the host or the environment. So those were some of the overviews of the different services that we offer. Like I mentioned earlier, we have 3 different package services for all of these different options, including whole genome sequencing, RNA-seq, metagenomics, and others. The value package, value preferred and express package, all of these packages include QC of your data, real-time project updates in your lab management account, as well as regular email updates as your samples move through various stages of the next generation sequencing workflow. We also offer PhD level technical support as in the initial stages of your project for project design, as well as anytime you need it in the during the workflow. This is going to be your most economical package, but if you have certain deadlines or if you are waiting to submit your particular data for grant, then we have the preferred and express options available, where we offer guaranteed turnaround times and a dedicated project manager for your particular projects. So that's all for my talk today. Please feel free to contact us at this email ID or phone numbers that are on the slide here. And I'll take any questions now. Thank you.
Well, thank you very much, Dr. Burla. It was very good and definitely very helpful and informative. Anybody, please feel free to ask questions either in English or Portuguese. If in Portuguese, we'll be glad to translate for you. Meanwhile, I have a couple of questions for you, Dr. Burla. And well, we all noticed that you guys have a very large technical platform with different equipments, as well as different setups for projects, right?
Mhm.
In that regard, I'd like you to comment on how important it is to have this kind of assistance to understand your project needs. Hence, having a conference for a technical consultation with you guys is important to define your project. Maybe you can talk a little bit more to our customers so they understand better how important that is based on the diversity of equipments and reagents that you have to offer.
Absolutely. As I mentioned, we have many different sequencing platforms as well as library preparation technologies that are available. And if you have a particular research goal in mind, I would highly recommend that we schedule a technical consultation call. The reason for this is sometimes, especially as somebody who is new to next generation sequencing, you may have a certain idea about what kind of service you need. But when you do discuss this with experienced scientists, we may be able to suggest a better approach or a faster approach or something that may be even more economical for you. So, for example, I have many of these technical consultation calls almost daily with different customers here, and the customers come to us with certain idea about what their projects are, but sometimes, especially if they are completely new to this particular field, we are able to suggest a different project design or something that they need, but a better approach to the entire project design, which really helps them. And the consultation doesn't even have to be very long. In pretty much 15 to 30 minutes, we are able to understand the particular project and recommend all of the downstream workflow that would set them up or set you up for success.
Perfect. I have a question from Tatiana. If I understood correctly, she's asking which are the limitation applications of RNA-seq?
That's a good question.
Yeah.
So RNA sequencing has many different applications. It is mostly useful if you are doing gene expression analysis in different types of samples. But if you let's say, it also depends on the particular your particular research end goals. So there are several different options depending on whether you're interested in messenger RNA or whether you're interested in mRNA and long non-coding RNA. But apart from that, if you have specific specific needs or requirements for long read technologies or isoform detection, it is going to depend a little bit more on your budget as well. So more than the limitations of the applications, I would say it comes down to the end goals of your project. The specific applications for RNA-seq are geared towards transcriptomic studies and gene expression or differential gene expression analysis. But if you are looking for something different, then we can certainly discuss more for your particular individual project and see if RNA-seq would be applicable or not.
Thank you, Tatiana. Please feel free to add anything to your question if it's not fully answered. I have another question for you, and you mentioned your metagenomic services, right? And and the predefined setups that you have to offer. But we also know that Genuis offer customized projects. Can you please comment on that?
Absolutely. As we talked about, with all of these services that I mentioned today are pretty much our standard workflow, but we are happy to accommodate any custom workflows that you specifically need for your particular projects. Of course, we would have to assess the feasibility of the assay at our end and check with our lab personnel to make sure that we are able to do it. But if it is something where there may be published papers out there, or if there is just a tweak that needs to be made to the standard workflow, we should be able to accommodate that.
All right. In other words, if somebody asks for a specific region or specific primer sets, you can actually do that and analyze the feasibility of that and the quality of that that primer setup, for instance.
Yes, we should be able to design custom primers for you.
Perfect. That's great. Any more questions for Dr. Burla? Let's take advantage of her expertise and her time. Nobody else? Well, I think that just an overall point of view for this kind of services is sometimes it is very hard to know exactly what kind of approach you need for your project, right? And I think that's the most interesting part of these technical consultations, and the value. That's a very interesting value for the service, right? And have you ran into many times where the customer had a request, and after this call, they completely changed their mind and understood that there are a couple of questions, that technical possibilities that they were not aware of, and things changed drastically. If you can comment on a couple of examples to illustrate that, it would be interesting.
Yeah, sure. So this happens more often than you would think, where the customer comes to us with a specific idea for their, for their particular project. For example, I had a recent customer who came to us with a specific amplicon sequencing project. But as we talked more, realizing that they initially were thinking about going with the Illumina short read sequencing, but as we talked more and realized what their applications were downstream, we actually recommended them to go with the PacBio long read platform, because some of the analysis that would be needed, and especially due to the longer size of the amplicons that they were working with, the long read technology made more sense. And ultimately, once the project was completed, they realized that that's exactly what they needed. So this happens quite a bit, where because there's a lot of different next generation sequencing technologies and services out there. Today we just talked about 3 services, but there's actually many more. So sometimes when customers read a particular paper and come to us with that idea in mind, and we ask for some of those resources and go back and read those papers, we are able to better understand what that earlier research that was published, what was done for that, and then able to recommend the exact similar approach rather than what they may have in mind when they were starting out.
Well, excellent. Well, first of all, Tatiana is thanking you for your answer. We have another question, a couple of other questions. One from Andrea. He's asking, which is the major bottleneck for projects, in your opinion?
So for successful projects as such, the couple of major bottlenecks that I would say would be, one is the amount and quality of DNA or RNA that you would be working with. We have certain, ideal requirements for both of these sample types when you're submitting them to us. And we are able to work with a very wide range of sample. Sometimes it does happen that customers do not have or are not able to submit the the amount of starting input nucleic acid that may be required for a downstream workflow. So in that case, we would perform we would able to still accommodate that and be able to take on that project, but it would be at best effort. So in that case, there may be some changes in the downstream workflow due to that. So the quality and quantity of the starting input is very important with next generation sequencing. So if you're working with a specific sample types or species, especially if you're interested in doing NGS, that is something that I would recommend optimizing before beginning this before using this technology.
Great. Just like to mention that these technical consultations, that, as Dr. Burla mentioned, can drastically change the service you request. It's free, so let's take advantage of that, right? So let's use their knowledge and expertise. We have another question from Christiane. She's asking if a customer simply wants to sequence a sample, standard sequencing, just because they don't have the equipment for that. You can also provide this kind of very simple services, right?
Absolutely. So the initial workflow that I mentioned, where we offer complete service from project consultation to sequencing and analysis, it is not necessary that you do all of the services with us. So if you only want to submit your pre-made libraries for sequencing, we can certainly do that for you. Depending on the read depth that you need, we can either sequence it on any of the Illumina platforms and also do the downstream bioinformatics analysis for you if it is something as simple as RNA-seq or whole genome sequencing. So definitely we can do those just those parts of the project for you as well.
Okay, thank you so much, Dr. Burla. I guess we don't have any further questions, at least on my end, for now. And everybody is thanking you a lot, and it was a real pleasure to have you here today. And on behalf of Synapse and our attendees here, I'd like to thank you for your time and the excellent presentation. Okay.
Thank you, everyone. It was a pleasure being here. Have a good day.
All right, everybody. Have a good day. Bye-bye.